One of the buildings here at the UC Davis Mind Institute that the public does not get to see is our Biosciences building. So today we are taking you on a virtual tour of the research done here by some of our Mind Institute researchers.
The research that I'm doing in my lab here at the Mind Institute is mainly focused on researching the molecular mechanism that are behind the lead to neurodevelopmental disorders. And these include mostly Fragile X syndrome, FMR1 associated disorders, autism spectrum disorder, and 22q deletion disorder. For the past 20 years, I've been working on Fragile X syndrome and looking at the structure of the FMR1 gene, which is the gene that is responsible for the disorder. So in the lab, we look at the methylation, we look at the structure of the gene, and we have been using this information also to develop some biomarkers that can help us to monitor the progression of the disorders. So we're looking at a spectrum of different things using all the most modern technologies and try to identify those pathways that involve genes that they might exacerbate and act as the gene modifier and help us to understand how this disease develops. And so we have identified a number of potential markers, and this is really exciting for us. So we are following up this research and to see and identify that now that we have, can we do something? Can we target those markers and normalize, rescue those abnormality in order to slow down the process or treat them? This has been a great actual environment for this research because it allowed me to collaborate with so many great investigators that has pushed the research in this place way ahead of the games. And we have made a lot of improvement, a lot of advance in the research of Fragile X.
The primary focus of my lab is to understand brain development in children with autism across the lifespan. To do this, we have several large-scale studies going on where we recruit children as young as possible, close in time to the age of diagnosis, around 2 to 3 years of age, and follow them across early childhood and adolescence. We bring these children in and we gather as much information on them as we can. So we collect behavioral data, medical data, we do blood draws so that we can look at their genetic profiles, their immune profiles, and then we also do MRI scans at the Imaging Research Center. So we bring all of this information together to try to identify these subtypes of autism. I'm very interested in looking at different parts of the spectrum. So girls with autism is one area of interest. Kids with autism and intellectual disability is another area of interest. Kids with autism and gastrointestinal problems. These different subtypes likely have different etiologies, and likely children will benefit from different types of treatments and interventions. The overarching goal of my research program is to try to use all of this information from the brain, from the behavior, from the biology, to identify different subtypes of autism so that we can better understand the etiologies that might be different and also develop individualized treatments for children.
In the Bauman Lab, we study prenatal risk factors for neurodevelopmental disorders, including autism and schizophrenia, and other neurodevelopmental disorders as well. So first, let me tell you why this is important. We know that women who are exposed to viral or bacterial infections during pregnancy have an increased risk of having a child who will eventually be diagnosed with a neurodevelopmental disorder. It's important to pause here and point out, though, of course, most women are exposed to infection during pregnancy, and most go on to have neurotypical children. Our goal with this line of research is not to add stress or anxiety to expectant mothers, but to understand why some pregnancies are vulnerable and others are not, and importantly, to provide women with evidence-based guidelines for how to manage infection during pregnancy. So let me tell you a little bit about how we conduct this research in my lab. We use preclinical or animal models which allow us to stimulate the maternal immune response in the absence of an actual pathogen. This allows us to understand how changes in the prenatal environment can alter offspring brain and behavioral development. We use a variety of tools in my lab, including behavioral phenotyping studies, neuroimaging where we can measure brain growth and development, and ultimately neuroanatomical studies where we can examine the cellular and molecular changes that are brought about by these prenatal immune challenges. Our goal again is to contribute to the understanding of which pregnancies are vulnerable, which time points during gestation are vulnerable, and how we can safely manage infection during pregnancy to reduce the risk of neurodevelopmental disorders in offspring.
The Mind Institute is all about collaborative interdisciplinary research. We have active studies going on not only here in our building, but in multiple locations on the UC Davis Health Campus and on the UC Davis main campus.
The research that my lab does, in line with the mission of the Mind Institute, is really attempting to develop novel therapeutics for rare genetically linked neurodevelopmental disorders and different diseases across the autism spectrum. And we do this through the use of cell therapies, gene therapies, cell and gene therapies, using new techniques like CRISPR-Cas9 to induce epigenetic alterations or regulate gene expression across a multitude of different model systems. So my lab really likes to focus on the human genome and developing targeted therapeutics using induced pluripotent stem cells that we can isolate from patients, that we can later turn to neuronal stem cells or neurons, the cells of the brain that are the most affected, as well as utilizing different transgenic mouse models of disease. And so mice that maybe contain the same genetic mutations or variants as the disease that we're focused on.
Everything that we do in our lab is the make or break for basic research moving into the clinic to do bench to bedside research. We test potential therapeutics for efficacy, meaning does it improve a problem that the person and or our model organism is exhibiting. We test for efficacy mainly by looking at what the disease's main symptoms are. We call these symptom profiles phenotypes. In neurodevelopmental disorder, they're almost all behavior. So for intellectual disability, we're looking at improvements in learning and memory or improvements in cognition. For any sort of motor disorder or impairments with hyperactivity or other motor related issues, we want to see that the compounds improve that. With ADHD-esque disorders, we want to see that the compounds can increase function. My lab mainly focuses on rare genetic disorders that are associated with intellectual disability, but also have a component of pediatric epilepsy. And so for that, our outcomes have to do with measuring brain activity through what we call an EEG. We look to see that these therapeutics that are designed by other collaborators at the Mind make the brain activity so that it's not so seizure susceptible, so that less seizures occur. And we also, most recently, have been looking at sleep outcomes because, quite frankly, in neurodevelopmental disorders, most families, their number one complaint is sleep dysfunction and abnormalities that are related to sleep. And so we think if we can improve sleep, that would be a major help to many, many families and improve quality of life.
The work that I do for the Mind is to try to develop molecular therapies for the treatment of neurologic disorders. So where there are other people that find mutated genes that are causing this disease, we're trying to do something about that. And we're trying to do something about that on the molecular level, the same level as the DNA sequences that are mutated. So once we know what the mutation is that's leading to a neurologic disease, we can try to develop some kind of molecular therapy that can either compensate for that loss, correct that loss, or in some other way get around the problem that's created by the genetic mutation. And we're trying to create these kinds of molecular therapies for a variety of different kinds of neurologic diseases. We do that work using advanced techniques like CRISPR-Cas and viral vectors like AAV vectors, and we design them in our lab just like this. And then we have to test them first in human cells to see if they're going to be productive for us. And then we might actually try to test it in a mouse model of the disease. And that's a very important step that we have to go through to make sure that the thing that we're working on will not just change the genetics of the mouse, but actually improve the mouse's symptoms. That gives us hope that if we were to eventually try that in a person, it would improve their symptoms as well. There's a number of other hurdles that we might have to do before we actually enter it into a clinical trial, but those are the first steps that we focus on the most in our lab.
This is just a sampling of the important research we're doing here at UC Davis Health, on the UC Davis campus, and here at the UC Davis Mind Institute.